Today we're going to talk about Hardy-Weinberg equilibrium, and this is the worksheet that either I gave to you online or I gave to you in class. And the first part of it is just to refresh your memory about the vocabulary and the concept of alleles. So there's 23 organisms that are homozygous recessive. So that means they have two recessive alleles. How many total recessive alleles is this? So each organism has two. So it's 23 times 2. So it's a total of 46 recessive alleles. Because alleles are your big A, little a. It's the genes that are on your chromosomes. All right, if there are 450 organisms that are heterozygous, okay, so if they're heterozygous, they have a big A and a little a. So it's a total of 900 alleles, but only half of them are recessive. So it ends up being 450 recessive alleles. All right, if there are 34 homozygous recessives, 25 heterozygous, and 55 homozygous dominant individuals, that means little a's versus big A's. This guy has 34 times 2, so that's 68. This guy has 25 recessives and 25 dominants. And that guy has all 55 of them have two homozygous dominants, so that's 110. So all in all, the recessive alleles are 93 and the dominant alleles are 135. So what I want to know is the allele frequency. That means how common is that allele in the population, and I want to know it for the dominant ones. So you take all your dominants, which is 135, divided by all the alleles. So that's 93 plus 135, which ends up being 135 over 228. And when you put that in the calculator, the frequency is 0.592. So right at 60% of the population has these dominant alleles. All right, in the same situation as here was the allele frequency for recessive alleles. That would be the opposite. That would be all the recessives divided by the total number of alleles, and that becomes 0.408. And this should add together and equal 1.0, because that's 59.2% and that's 40.8%, which would give you 100%, because those are the only two alleles, so that makes it 100%. The next part of this is for us to kind of understand what Hardy-Weinberg equilibrium is. Hardy-Weinberg equilibrium is an equation that helps us measure whether or not a population is evolving. It's a very hard measurement to make, so we can use the equation to hypothetically determine if there is a change in the alleles in a population. And if the frequency changes or a new one gets introduced or anything weird happens, then we know we have some evolution going on. Now, Hardy-Weinberg also has to take into account when there is no evolution, which is pretty much non-existent, but there are some populations that don't evolve that much. All right, but there are 5 rules for Hardy-Weinberg equilibrium to occur, and equilibrium means no evolution, okay? So to have no evolution in a population, you have to follow these 5 rules, and they're very hard to follow. So the first one's okay, large population. Okay, so if you have a large population, you're less likely to have a lot of evolution because there's a lot of variety. As your population gets smaller, we're going to have increased amounts of mutations or inbreeding or things like that. All right, no migration. No new organisms moving in, no organisms moving out. Now, moving in is better because it increases variety. So immigration with an I equals increased variety, which could cause a change in the allele frequency, which would cause evolution. And then immigration with an E means moving out, and that decreases the variety, which leads to possibilities of inbreeding, which causes even more problems. Right? The third, and these are in no particular order, they all have to be met. So is no natural selection. So that means that one organism's trait is not better than another organism's trait. So like white fur is not better than brown fur, or being big is not better than being small, or vice versa. So absolutely no natural selection. And that one's a very hard one to accomplish because the environment is constantly changing, and so therefore your fitness would constantly change. We have to count for no choosing, is what I'll call it. It's called all random mating. So females are not choosing males based on any particular traits, and males are not fighting over females. So there's no choosing going on. So then there's no preference. And if there's no preference, then there is no selection. So with this process, this would be more like with animals that lay eggs and then the males fertilize them or something like that. There's absolutely no choice going on. They just combine their egg and sperm, usually in a water environment. All right, and the fifth one, no mutations. So your DNA cannot mutate. There are some organisms whose DNA doesn't mutate rapidly, but for the most part, mutations are random and they do occur. And so this is also a hard one not to break. Okay, so you have to know those five because you need to know what has to be true in order for a population not to evolve. All right, and then from your formula sheet, I want you to pause the video. I want you to find the formulas for Hardy-Weinberg equilibrium, and I want you to write them down because I want you to see where they are on your sheet. Formula sheet will tell you P plus Q equals 1, and then the formula sheet will tell you P squared plus 2PQ plus Q squared equals 1. Okay, on the flip side of your paper, there is, a table for you to fill in what everything stands for. I'm going to write it over here, but I do want you to take it and I want you to fill it in on your table. Okay, so these are two different equations. So they stand for two different things. This stands for alleles. So that's literally the letters. And this stands for the actual organisms. Okay, and so I'll explain what that means right here, and I'll write it in a different color. So P in this first equation stands for the the frequency of dominant alleles. Okay, not organisms, not individuals. That's how many total dominant alleles are in that population, which would come back up here because remember now, some organisms have 2 recessives, some organisms have 1, some of them have none. So these are the allele frequencies. All right, and then so therefore Q would stand for the frequency of recessive alleles in the population. And these are going to be decimals. So earlier we had 0.592 was our frequency of dominant alleles. So that would have been our P. And then our Q was the frequency of recessive alleles. So that would have been 0.408. And then 0.592 plus 0.408 should add up to 1. All right, so over here in the second formula, this stands for organisms that are homozygous dominant. So this is the frequency of homozygous dominant individuals in the population. So that's actual numbers of organisms, or the frequency, the percentage. This stands for heterozygous, the frequency of heterozygous individuals in the population. And this stands for homozygous recessive. So this is the frequency of homozygous recessive individuals in that population. All right, so we're going to actually use both formulas to figure out some math, but they're two separate formulas. So these two add up to 1, and these 3 add up to 1. These are alleles, these are organisms, these are frequencies, and these are frequencies. So they're always going to be decimals because they're all going to add up to 1. All right, so the second page, you can fill in that top part. But I gave you an example, and it said gray body color is recessive to black body color. So I'm going to do big B is black, little b is gray. All right, and it says if a population of flies has 567 gray flies and 342 black flies, use the Hardy-Weinberg equilibrium formula to calculate the following amounts. Okay, so the only one we know for sure is the flies that are gray, okay? The flies that are gray, we know that they have to be homozygous recessive because black is the dominant trait, or the dominant allele. And therefore, if you have a big B, we don't know if these black flies here are big B or big B little b because they physically would look the same to us. So if we saw a black fly, we couldn't say for sure oh yes, definitely they're homozygous dominant or they're heterozygous. We would just know that they have at least one dominant allele. The gray flies are pretty simple. We know for a fact that they have those two. So on this chart, they represent which one of these letters? Okay, so they represent our Q square because our Q square represents the frequency of organisms in the population that are homozygous recessive. So we have to figure out the frequency of those flies, and that means we take the total number of flies that are gray and divide it by the total number of all the flies. So we have 567 plus 342. So that adds up to how many flies? 909? Okay, so we have 909 flies in this population. 567 of them are gray. So then we know we take that 567 divided by 909, and we're going to get 0.3. Oops, sorry. We're going to get 0.624. Okay. So that's just the division, and I'm going to leave it like that as a decimal. So 62% of the population is gray, and that makes sense because they're the most numerous of this example. And then the actual number we already know is 567. Okay, so we don't know, but the P squares represent the ones that are homozygous dominant, so they're going to be black. And then 2PQ represents the ones that are heterozygous, and they're also going to be black. And then these are the gray ones. So little news flash, always start with your Q squares. Your Q squares are the ones that physically have that recessive trait, and so you know that they have to be homozygous recessive. Okay, so from there we're just going to do some math. So we're going to work this thing like a little puzzle. So we're going to start here. Our Q square is 0.624. Q is right here. So if I take the square root of Q squared, it gives me Q. So in your calculator, take the square root of 0.624, and you should get 0.790. All right, very simple. Then we're going to use the formula. I'll do it right up here so you can see. We know that P plus Q equals 1. We just found that Q is 0.790. So in order to find P, we just subtract 0.790 from both sides, and we find that P is equal to 0.210. So then I can put that there. All right, so I'm just going through and using my formulas to do math. Now to find my homozygous dominant, all I have to do is take that number I have for P and square it. So 0.210 squared in your calculator is 0.044. And then, final step to find the frequency of the heterozygous organisms in my population, I literally do 2 times P times Q. So 2 times 0.210 times 0.790. And just do that in your calculator, and you're going to get 0.3318. All right, so those are all frequencies. Now I'm going to draw a dividing line here because we're going to do 2 different things to get the actual numbers. All right, so down on the bottom, these are the individuals we have 909 individuals. Up on the top, these are the alleles that the individuals in this population have. And since there are 909 individuals, each one of them has 2, so it's 18 alleles. So 909 individual flies, 1,818 alleles. So in order to find out the actual numbers, you take that times 1818, because that's the frequency of the dominant allele in the population, times the total number of dominant alleles in the population, and you are going to get 381.7. Okay, so with your calculator, and then we're going to round that to 382. So about 382 dominant alleles. Same thing here, set 0.790 times the total number of alleles. And that's going to give me 1436.22. So that's going to round to 1,436 recessive alleles. Those aren't individuals, those are just alleles. Now to do the individuals, we take this bottom equation, because these 3 go together, and we multiply that by 909. Okay, so now 0.044 frequency of homozygous dominant times 909 gives us like 39.996. So about 40 of the flies that are black are homozygous dominant. Then 0.3318 times 909. In your calculator, you get 301.6062. So on average, about 302 of the flies that are black are going to be heterozygous for the trait. So in general, this is how you estimate the allele frequencies, and you estimate the actual number of the different genotypes based on using the Hardy-Weinberg equilibrium equation. All right, so the next example, I want you to try and do it. Stop the video, try and calculate it. If you've already done it, then you can check yourself. But we're going to do this one. I'm going to do it pretty quickly so that you can follow along. All right, here, white wool is dominant to black, so I'm going to make big A white, little a black. Okay, so my Q squares are my homozygous recessives, my 2 PQs are my heterozygous, and my P squares are my homozygous dominant. Draw a line here. These are my dominant alleles, and these are my recessive alleles. All right, so in the population, there are 900 total sheep. So I know that I'll take this frequency and multiply it by 900. If there's 900 organisms, that means everybody has 2 alleles, so that would have to be 1,800 alleles. And that's how I'm going to get my actual numbers. All right, always want to start with the organisms that physically have the recessive trait, because those are the ones we know for sure their genotype. And in this situation, there are only 9 of them. So 9 out of 900 are black. Okay, so these would be white, and only 1 black. Okay, 9 out of 900, which if you divide is 0.01. Okay, and I know for a fact that there's 9 individuals here. All right, same thing, we're going to hop through. So this is Q squared, square root, we can find Q. And the square root of 0.01 is 0.1. Then we can use P plus Q equals 1 to find out that if Q is 0.1, then P has to be 0.9, because 0.9 plus 0.1 is 1.0. Then to get P squared, we square 0.9. We're going to get 0.81. And then to get 2PQ, we do 2 times P times Q. So 2 times 0.9 times 0.1, and your calculators, you should have gotten 0.0162. All right, so draw my little dividing line here so you don't mix those numbers up. I'll start at the bottom. So 9 of the sheep are black. The rest of 891 are white. So we want to figure out how many are homozygous dominant and how many are heterozygous. So we multiply the frequency times the total. We get right at 729 would be homozygous dominant for white. And then only about 15 are going to be heterozygous for that trait. Then for the alleles, there should be a lot of dominant alleles because 90% of them are dominant, which is going to be this times this, which is 1,620. And then you do this times this, which is 180. Okay? And that's how you do that Hardy-Weinberg equilibrium. Next page, you have another example. I'm going to give you a little bit of heads up on it. This one has a blend, so it's an incomplete dominance. So you have tan, you have golden, and you have white. I'm just going to go ahead and tell you your P squares are your tan, your 2 PQs are your gold ones, and your Q squares are your white ones. All right, so I want you to pause the video, see if you can come up with the data that would go in the chart, and then you can come back on and I'll fill it all in for you. All right, so for the chart, you have 172 that are white. You have 482 that are golden and 234 that are tan because they've already told you that. But you need to find the frequencies so that you can find P and Q. So we always start with our recessives. In this situation, we can start with our Q squares. So it's 172 out of 888, which gives us a frequency of 0.193. Then we go up to Q, we take the square root of that, and we get 0.439. Then we take the formula of P plus Q has to equal 1, and that lets us know this is 0.561. All right, you don't need to do the frequencies, but if you did, this would be 0.264, and this one would be 0.543. All right, so because these are already given to you, which is nice. But here, we can do the actual number. So there are a total of 888 bees or whatever these organisms are. And so if I want to find the number of alleles, every one of those organisms has 2 of them. So that actually is 1776 number of alleles. So to find the frequency of the allele, dominant allele times 1776 comes out to about 996.336. So we're going to average that to about 996 straight up. And then you multiply those two and you get 779.664. And I'm going to round that to 780. Okay, so those are your allele frequencies for this population of animals. All right, same thing. Read this one, see if you can do it, and then read the bottom one and see if you can do those. Pause the video, work them out, and then come back and see what the answers are. So with this example, 4% of the population has sickle cell, and that's homozygous recessive. So that already is a 0.04 frequency. So that's our Q square. So we can go from there, take the square root of it and find Q. Right, and the square root of that is 0.2. Then 0.2 plus what equals 1.0. So that lets us know that our dominant allele frequency is 0.8. And then what they want is, or excuse me, what I want is calculate the percent of individuals who enjoy the protection from malaria as carriers. Okay, so carriers are your heterozygous. So in this question, I didn't want everything. I just wanted to know who isn't sick but is protected, and that's going to be your big A, little a. So the frequency will be 2 times 0.2 times 0.8 because that's 2 PQ. And so that turns out to be 0.32. So in that population in Africa, we can estimate that if 4% of the population is born with sickle cell, about 32% of the population actually is heterozygous for it and highly protected. Down here with the red hair, I'm going to do big B equals brown, little b equals red because I told you that red is recessive. And then I told you that 38% of the population has red hair. So that gives you Q automatically. So if I can find Q square, which is where we always start. So Q square is 0.38. So to go to Q, I take the square root, which is 0.616. And then I use P plus Q has to equal 1 to find that this is 0.384. And then I square P to find that this is 0.147. Sorry, that's very ugly, 147. And then I use 2 times P times Q to get 0.473. All right, to get the actual number, we have a total of 14,500 individuals. Therefore, they all have two alleles, so that would be 29,000 alleles. So if 38% of that population has red hair, we do times 14,5, and we get 5,510. Here times 14,5, we get, I'm going to round it to a whole number. It's going to be 6,859. And then here times the total population, and I'm going to round that to 2,132. All right, so that's our estimated total actual individuals. So this many people would have red hair, and these people would have brown hair, but these would be our homozygous dominants. These would be our heterozygous. And then in this, these are our allele frequencies. So since we have 29,000 alleles in that population, we would estimate that 11,136 would be the dominant alleles, but 17,864 would be the recessive alleles, because there's a lot of people with those recessive alleles. All right, and last but not least, we're going to do a chi-square to test whether or not our population is evolving. So I asked you to go use the data from the question about the gray and black flies, and we're gonna revisit that population. We're gonna pretend like it's 5 years later, and we've returned to the area, and we're gonna recount the flies, and we're gonna get their new phenotypic frequencies. All right, so now in our 5 years later, we have 984 black, and then we have 872 gray. So I'm gonna do big A, this should be genotype, and then I'll do big A, little a, and I'll do little a, little a. All right, so right now, 5 years later, I come back and I count, and I have 872 gray flies, right? That's the only thing I can kind of start with right now. I need to go and figure out what my expected should be if this population has not evolved. So I don't use the actual numbers from the original test. I'm going to use their frequencies. All right, so from the fly data, look back in my paper, we found that population 5 years prior, 0.044 were homozygous dominant, 0.3318 were heterozygous, and then 0.624 were homozygous recessive. All right, so we use their frequencies, but we don't use their actual numbers, okay? Because this is a whole new number of flies. So in this fly population, 984 and 872 adds up to a total of 1,856 flies. Okay, 1,856 flies, okay? So if this fly population has not evolved, then they should be in the same frequency. So we'll multiply these frequencies times 1856 to determine what we should expect if they're in the exact same frequency. And when you multiply that, I'm going to leave them as decimals so they'll be beautiful. I'm going to get 81.66 are expected to be homozygous dominant. 615.82 are expected to be heterozygous, and then 1,158 would be expected to be homozygous recessive if they stayed in the same frequency as they did in the 5 years ago. Well, we observed 872 gray flies, so we know that there's some change going on. So we got to use those numbers and figure out our frequencies for these two. All right, so 872 are our Q squares. So it's 872 out of our total population. So our Q squares are 0.8. Oops, sorry. Gotta get my calculator out. All right, so our Q square is 872 divided by the total number of flies, which is 1858. All right, so that is Q square equals 0.469. All right, so now I'm going to take the square root of that to find Q. And I'm just going to do 0.469, not the whole number. So I find that Q is 0.685. And then I do 1 minus 0.685 to get my P. So P is 0.315. Whoops. So now my P squared is going to be 0.099. And therefore my 2PQ is going to be 2 times 0.315 times 0.685. So 2 times. So that frequency is going to be 0.432. All right, so my 2PQ are my heterozygous. So that would be 0.432 times my total population, which is 1858. So 0.432 times 1858 is 802.656. That's what we should expect. And then my homozygous dominants are my P squares. So that's 0.099 times 1858. So 0.099 times 1850 is 183.942. Okay, so if they stayed exactly the way they were 5 years ago, those are our expected. So now we're going to do our O minus E. So our chi-square is observed minus expected squared divided by expected. So O minus E, and then you square it, and then you divide it by the expected to get your chi-square value. So I'll do these in blue, and you do them for each number. So I'll do 183.942 minus 81.66, square it, and then divide it by 81.66, and I get 128.11. Second one, O minus E, 802.656 minus 615.82, square it, and then divide it by the expected. And it's about 56.68. Little bit closer to what was expected. And then last one, 872 minus 1058.14 squared divided by the expected. And that would be 70.70. All right, so now that's our total chi-square. We add them all together to get the total. So our total sum, or sum of the chi-square, is 128.11 plus 56.68, and then plus 70.7. So our total chi-square is 255.49. All right, it's a big number. We're definitely going to see some evolution, but to support our evidence, we use this calculation, our degrees of freedom. There's 3 options. So our degrees of freedom is number of options minus 1, so 2. And then we use our table. When we look at our table, the critical value we have to exceed is 5.99. All right, so the null hypothesis says that there is no evolution. No evolution has occurred. So do we accept or reject that null? We have to reject that null. Okay, so we reject the null because our chi-square value is 255.49, and it is greater than the critical value of 5.99. So this shows that definitely something is going on, and we would have to investigate further and figure out why all of a sudden has black become a more popular number than we had anticipated. Could be an environmental change, it could be a mate choice preference, but definitely it is not in Hardy-Weinberg equilibrium.